it's really amazing to me looking at this gallery, and I actually was not participating in you setting it up. And at one point towards the end, I decided to surprise myself because you were being very much like an artist. You kept changing your mind of what you were going to do, which I loved. I thought that's really interesting. You're kind of allowing yourself to keep changing your mind. So I wanted to surprise myself, and I did. I mean, I found, I don't know what I was expecting, or maybe I did not have, I didn't want to presuppose what I was going to see. But when I walked into the gallery, I was so pleasantly surprised at the kind of pleasing aesthetic that even if I didn't understand very much, it would intrigue me to look. And that's really what any artist would want to do. And so you could, if I said to someone, she's a conceptual artist, there's no question that people would say, oh yeah. Do you know what I mean? So it doesn't seem to be something that's not
That a
fitting in
scientist
the
did.
gallery, or no, it's just, it kind of proves what I keep thinking, and that gives me incredible satisfaction that this kind of process can help you, but also can help others perceive the scientific process in a different way, and then also inform us in a way that really makes us curious. I truly want to go to this place now.
Oh, yeah.
I've heard about it, and it's come and gone. Certainly when I did my research, I heard about it.
Yeah.
But this made me really want to go. It kind of brought it home in a tangible way, and not just something I'd learned or read about. So I have to commend you. You did pretty good.
Thank you.
This is a good show.
That's great.
Thank you. So we're going to open it now to the class, and because we have just one camera system, I guess
can
I'm
repeat
going
the
to,
question.
Yeah, repeat the
Yeah.
question. Perfect. So there must be some questions from you guys. A quiet pause.
Who inspired you when you just started getting into micro?
Who inspired me?
Or
Bacteria inspired me. I think they're just awesome. I've been really into microbiology since I was in college. So, yeah, I think everything I learned, I was just like more and more fascinated by how these bacteria do things, like, and how they sort of have these. I'm now really interested in like communities of bacteria, like the ecology and stuff, and sort of like how bacterial sociology is something I'm very interested in, how they communicate with each other, and how they interact with us and with an environment. So I'm interested in how these kinds of things that are so microscopic and so like, I mean, the word I'm thinking of is liminal, which means like in between. Like they're sort of in between us and the air, and in between like a lot of different kinds of things. And I'm interested in bringing those in between things out as a scientist and as an artist now.
You said you were a biological engineer as well. What do you do on the engineering side?
So my research in grad school, I did a lot of work with like biofuels. I started out trying to sort of engineer metabolic pathways inside of cells to produce fuels. I did then different kinds of projects in this kind of microbial sociology way, and thinking about symbiosis, so how organisms live together, and especially endosymbiosis, how bacteria live inside of other things. So like the mitochondria or the chloroplast are symbionts of bigger cells, and so I was trying to engineer organisms to live inside of bigger cells. So I did a lot of stuff like that. So actually the last two sort of components of the project are in that kind of more genetic and like biotechnology world that I sort of come from. So the lab I'm in now is definitely like a microbial ecology lab, but the biotechnology and the genetic work is where I came from. And so the little test tubes are genomic DNA that I extracted from the strains, and I used to identify the species that I had here. So like using, I think of it sort of as an abstraction, and I've been actually talking over Facebook with somebody who came that I didn't actually meet, who was like writing me these like messages on Facebook about like my use of the term abstraction, which I now am like taking much more seriously from the, but anyway, so this idea that, yeah, that the genetic, the genome is an abstraction from the organism, that like you can get a lot of information about the organism from looking at its genes, but you're not going to get the whole picture. You're seeing a certain like a fragmentary kind of knowledge. So that's that kind of abstraction there. And then the last one of the strains I found, in doing some research on it, I found that it has a really high concentration of a molecule that's used in dietary supplements that's really expensive. And so there's this part of my field is people like go out into the wilderness or to the sea often and like look for genes in the ocean that may produce molecules that are expensive. It's called bioprospecting. It's like prospecting for gold, but in this case the gold is actually the sequence of an enzyme that you can use in biotechnology. So I was really interested in this idea of like the Salton Sea becoming this like very abstract resource for genetic information, and also people have been kind of like treasure hunting around the Salton Sea, because when it was a lake that was connected to the ocean, there's this rumor that a Spanish ship full of pearls went in, and then the sea dried up and they couldn't get back out, and so it's like that there's a ship in the desert full of pearls somewhere, and so there's these treasure hunters who are trying to find it, and so there's this really interesting like hunting for treasure in these kinds of weird spaces, and sort of like this idea of extraction from
It gives me this visual of all the white dead fish bones with pearls.
Yeah.
Wouldn't that be amazing for kind of painting?
Yeah. That would be interesting, definitely. Yeah.
Are these images of the Salton Sea?
Yeah. Yeah, so that's that's like pretty zoomed out. The sea is like 35 miles long. You can see, so it's right along the San Andreas Fault, and so you can sort of see where that fault is, like underneath this like line where the agriculture and the sea goes. And I thought I liked sort of using this map as sort of like a guide to anchor people to like where we are and to sort of see the desert and the mountains and this agriculture that surrounds the area. And then I was just like totally fascinated by the way that there's like these sparkles in the water from the artifacts from the satellite images that are so beautiful. And I haven't been able to find them in other bodies of water as I'm like looking around in Google Maps. So I wanted to sort of blow that up and make, again, like an abstraction where like you're seeing the water in a different way and it's sort of alien through the sparkles that look like sort of galaxies.
Did you search through NASA maps, maybe?
I didn't look.
Ah, you should look.
I should look.
NASA maps are pretty incredible and high resolution, and we paid for them with our tax money, so
Oh, yeah.
you can just grab them. But really, you may find something that's much more accurate.
Oh, yeah.
And unfortunately, because of the sequester, that program's kind of stopped.
Oh, really?
But there's still the maps there.
Okay.
So
I
you
should check
take
it out.
a look. Yeah.
That would be really fun.
And anybody here, if you go into NASA satellite images, you'll be amazed at the kind of incredible satellite images of maps you can find. Any last question? Yes.
How do you know that some kinds of bacteria can affect how you smell, how people smell?
Oh, because our bodies are covered in bacteria, right? And they the smells of body odors are actually mostly due to the metabolism of the bacteria. And like when bacteria chew up like oils in your skin, those products, like the breakdown products of oils, are what smells like. And it's that's why it's so similar to the like body odor and cheese odor are similar, because it's like the breakdown of the oils and fats in milk are what makes cheese smells. So like butyric acid, for example, I don't know if you're a chemist or like taking orgo, OChem, the like butyric acid, isovaleric acid, like all those molecules smell like really sweaty, and they come from like butyric comes from butter, right? Like rancid butter, and like it's all the same kinds of like metabolic breakdown products of these bacteria. So, and between your toes, you have different strains that are also related to bacteria, and like cheese that smell like feet, then like a different set of sort of molecules. So there's a huge literature that I was very, very fascinated by, obviously, and sort of digging into where people would like take these different microbes from the body and then like look for what made the different smells and like try to isolate those molecules.
So maybe at the end of the class you can tell the students about the Human Microbe Project.
Oh yeah.
And how many microbes and different bacteria and all that. I mean, it's like a whole nother society in each one of our bodies, a whole network that you have to kind of think. We think differently about ourselves once we know this, right? So
I think
the
so.
Human Microbe Project, just give them a little taste of that.
Yeah, so the human microbiome, so like there's the genome, which is like all of your genes, but it turns out your genes make up only a sort of small-ish fraction of all of the genes that actually are in your body, because for every one human cell, there are 10 bacterial cells, at least, that are either on your skin or in your gut. Your gut has 3 to 5 pounds of bacteria that are all sort of interacting with each other, with you. They're controlling your immune system, your digestion in ways that we just really don't fully understand. And there's really, really interesting evidence showing that there's a connection even between the microbiome and your mood and personality, and like so many different kinds of connections that are really fascinating and like just beginning to understand or beginning to explore. So the Human Microbiome Project was just to sort of get a census really of like the kinds of species that can be there in different parts of the body. So they had like, I'm not sure how many people they did, but probably like around 100 people, and they sampled from different body parts from 100 different people, and they found like huge diversity like on individual people and then between individual people. So there's, I can't remember, there's like alpha diversity and beta diversity. I think alpha diversity is like on a single person that you have like 100 strains on your skin or something. But then there's beta diversity that's between different people. There's a lot of diversity too. Like my 100 is going to be completely different than somebody else's 100. And like how those hundreds are are different from each other, and that you can associate them with different things like obesity, diabetes, like tons and tons of stuff that people are just starting to sort of pick out. So
Fascinating.
it's fascinating. Yeah.
All right, let's give her a big hand.